Hello everyone. Thanks a lot for watching my talk. For those of you who might be wondering about the picture in the background, it's a view from the Mendota Terrace here at Madison, Wisconsin. It's different from the Monora Terrace where ISMB will be held, but it's still a view worth capturing. And I hope everyone can take a glimpse of it while they're here attending the conference. Coming back to my presentation, it's titled Hunting for Hidden Chemical Treasures, and my motivation to the presentation is to just ignite a curiosity about or a view of appreciation about sponges and other deep sea organisms and the treasure troves of natural products that they harbor. So without further ado, let's get into it. The hero of our story is the sponge Forsythia. And just to highlight what the sponge looks in its own natural habitat, I want to show this clip which was taken when the sponge was actually extracted near the Gulf of Mexico. And it's just cool to look this single sponge sitting in the whole ocean, just having chemical compounds. So why do I like this sponge? Well, this sponge produces a compound called Lasonolide A. And the reason this compound is so cool is because it's been shown to have potential anti-cancer activity in nanomolar concentrations. And if that wasn't enough, it also shows a really unique mechanism of action which is super cool for anti-cancer drugs. So what's the problem? Well, the supply of this compound is limited. The sponge is the only known natural producer of this compound. And as you can imagine, we can't just keep scavenging these sponges just to extract Lasonolide A. It's just not sustainable. Well, someone might say, why not call the organic chemist in the house and ask them to produce this compound? Well, yes and no. People have tried this and it's a pretty laborious process and the yields are not great. So what do? Well, it's been shown that whenever sponges are producing a natural compound, it's actually the symbiotic bacteria that's associated with the sponge that's producing it and not the sponge itself. Furthermore, if you look at the structure of Lasonolide A, the beta branches and the tetrahydropyran ring suggest that it's being produced by a trans-city PKS pathway, which are more commonly found in bacteria. So, hypothesizing that Lasonolide A is being produced by a bacterium, we sequenced the entire sponge metagenome. So we did shotgun metagenomic sequencing, took the reads, assembled the reads into contig, and then pinned the contigs into metagenome assembled genome. And after binning, what we got was a cool plot that looks like this. So it's a really complex graph, so let's break this down together. The two axes are just dimension reduced axes of a 512 chemofrequency table. And each dot on this graph is a contig. And it's been known that contigs belong to the same genome, would have similar chimeral frequency or nucleotide composition and would cluster close to each other. So all the contigs clustered together form one bin or one genome. And by analyzing this data, I was actually able to identify the contig having the Lasonolide gene cluster, which lies here. And further analysis led to identification of the genome that actually has this contig, which is here. And as I just mentioned, that contigs belong to the same genome would cluster close to each other. But this is not the case here. These two are really far apart. So what's happening? Well, we think that last VGC is being horizontally transferred. And when a gene or genes are horizontally transferred, the nucleotide composition is different. And hence, that's why they're separated. And the reason I think it's horizontally transferred and not just a annotation or misclassification is because we verified that this contig actually belongs to this genome using assembly graph, mapping Bayesian reads, and even using PCR. So we know it's actually connected. So now we know it's horizontally transferred. Let's take a deeper dive into what this last PGC looks like. So this is the Lasonolide gene cluster. And I actually went through each gene by gene, annotated each gene, and was actually able to identify the whole biosynthetic scheme for the synthesis of Lasonolide A. And this actually turned out to be trans-city PK pathways as we hypothesized. So that was pretty cool, and so a pretty excited moment to see that this contig is actually producing Lasonolide A. Another thing that I have not mentioned about Lasonolide A or LASPGC in particular is that it's not just present once, but it's repeated 3 times. And the first hint of these 3 repeats came from 3x coverage of LASPGC compared to its host genome. And to validate if it's actually repeated thrice, we did some assembly graph analysis and verified these connections using PCR, so we know it's actually repeated thrice. Furthermore, once I knew it's repeated 3 times, I was curious to see if there's actually any differences between these 3 repeats. And lo and behold, there were some. There were 3 insertions and 2 single nucleotide polymorphisms between the 3 repeats. Fortunately, the 3 insertions were in a multiple of 3, so they were not causing any frame shift mutation. Furthermore, the 3 insertions were not coding for any PKS domain as well, so as we know, as of yet, they're not affecting the translation of Lasonolide A. Regarding the SNPs, SNP1 is coding for the same amino acid after the substitution, that's tyrosine, and SNP2 is actually outside the coding region, so it's not coding for anything at all. So I've been talking a lot about LASPGC, but what about the bacterium that actually harbors this content? Well, this bacterium actually belongs to a novel genus under the phylum Verrucomicrobiota. And the moment I found out it's a new bacterium that's never been found again, I was like, yeah, let's name this thing, because as scientists, we all thrive to name stuff, right? Like, I found this new thing, let's name this. So I named this Candidatus thermopylae lasonolidus, and I'm gonna show you why. Well, Candidatus, because it's still uncultured, I just found this using shotgun metagenomic sequencing. So Candidatus is there. Thermopylae is actually a tribute to the 300 Spartan hoplites who fought about 1 million Persians at the Battle of Thermopylae. So there were 300 Spartans, and our BGC is repeated thrice. Furthermore, the Spartans fought against the Persians to protect their kingdom, while our bacterium, we hypothesized that our bacterium is producing Lasonolide A to protect the sponge, which is its home, from any predators. So that's the analogy there. And if you still don't remember Thermopylae, I hope this image might remind you of it. It's from movie 300, which is based on a battle. And if you've not watched it, I highly recommend watching it. Finally, Lasonolidus, because it's producing Lasonolide A. So I've been blabbering a lot for the last 8 minutes or so. So let me summarize what I've said. Lasonolide A is a potential anti-cancer drug candidate that's produced by Candidatus thermopylae lasonolidus. Lasonolide A is produced by a trans-AT PKS pathway called LASPGC. LASPGC is horizontally transferred, and it has 3 repeats, and those 3 repeats have some differences. And my talk would be incomplete without thanking all these amazing people without whom this work would never have happened, the whole Quan Lab, as well as my dear collaborators. Furthermore, if anyone is interested in collaborating, anything regarding bioinformatics, I'm more than happy to talk to. Please contact me, just ping me, I'm happy to chat. And the story that you heard today is just a really small snippet of our entire story. It's on BioArchive. If you're interested, feel free to read it and let me know your feedback. Thanks a lot.